Meiosis. Meiosis is the special type of cell division in which number of chromosomes in the daughter cell is reduced to half as compared to the parent cell. You can see this is a diploid cell which would be converted into haploid cell via the process of meiosis. That diploid cell have double number of chromosomes and haploid cell have half number of chromosomes. This is the general overview of the meiosis. In interphase, the DNA would be replicated. So after the interphase, homologous chromosomes undergo meiosis 1 and meiosis 2 to form the 4 daughter cells which are haploid. So meiosis 1 is also known as reduction division because here the homologous chromosome number is reduced to half. This is the prophase 1 of the meiosis. This is the prolonged stage. It is the prolonged stage as compared to other phases of the meiosis. Prophase, pro means before, so this is the phase before the other phases. This phase is different from the prophase of mitosis because here the homologous chromosomes are present which are in contact and form the synapse with each other, and here the crossing over also occurs. In mitosis, the chromosomes are just aligned. In prophase of the mitosis, there are no homologous chromosomes. In metaphase 1 of the meiosis, what happened? That the homologous pairs of chromosomes would be aligned along the center of the cell to form the metaphase plate. These spindle fibers will help to align these chromosomes along the metaphase plate. Next, cell enters in anaphase 1 of meiosis, where the spindle fibers will start pulling the homologous chromosomes toward the opposite poles. You can see in the telophase 1 of the meiosis, the cytoplasm starts to divide. So here the cytokinesis starts and nuclear membrane will start forming. Centrioles would be replicated. So here the 2 daughter cells are almost prepared. Next is the cytokinesis 1 of the meiosis, where the cell would be divided into 2 haploid daughter cells having their own centrioles, their own nucleus, and their own cytoplasms. Next, the 2 daughter cells which are formed through the meiosis 1 will enter into the meiosis 2, which is just like the mitosis. So this is the prophase 2 of the meiosis, the first stage where the cell enters. These 2 daughter cells will enter. And we know that in prophase 2 of the meiosis, chromosome will become pair up, chromosomes become thicken and shorten and become visible. Centrioles will move toward the opposite poles of the nucleus. Nuclear membrane starts disintegrating. Nucleolus will also start disappearing in prophase 2 of the meiosis. Next is the metaphase 2 of the meiosis. In metaphase 2, the chromosomes start aligning via the spindle fibers. So here the chromosomes will be aligned at the equatorial plate, which we call metaphase plate. Next is the anaphase 2 of the meiosis, where the spindle fibers would contract, pulling these sister chromatid toward the opposite poles. So here the chromosomes get pulled apart. Next is the telophase 2 of the meiosis, where the chromosome will become uncoiled. They will be unfolded. Here the spindle fibers would be disintegrated. They would be removed, and centrioles would replicate. You can see each daughter cell would have their own centriole, and the cell starts to divide here.